Thanks. So I'm Ian. I'm a third-year student in the P3 program. The title of the talk is Computational Classification of Phenologs Across Biological Diversity. So one of the main topics that my research is focused on is biological ontologies. And so I'll jump in with just a quick introduction to what those are and how we use them. So ontologies are hierarchically ordered graphs of terms from a specific vocabulary. And we can use them to infer implicit information and reduce sparsity of a data set. And the small portion of an ontology that I'm showing on this slide is some terms available in the gene ontology, which if you've read things like enrichment studies is usually the ontology that you've heard of. So an example of how we can use this to reduce sparsity of data, say we have a phenotype like leaf senescence occurs at time t. We can annotate this phenotype with a term from the graph, like the one that's lit up in red, leaf senescence, and then infer by going up the hierarchy towards more general terms that leaf senescence is a type of plant organ senescence, which in turn is a type of aging, and keep following it up the hierarchy until you get to, most generally, it's just a type of biological process. And so this is a more information-rich look at what was originally being described just in the text for that phenotype. Another example of the utility of looking at phenotypes this way is we can get a quantifiable measure of how similar they are to other phenotypes. So if we have the second one, increase in replicative cell aging, we can do the same process and we can see this second set of nodes in the ontology lighting up, and the overlaps are shown in purple. So if we want to ask how similar they are, we can say there's 3 overlapping and a total of 7, so the similarity might be 3 out of 7. So our lab is concerned with building computational tools that help biologists answer questions or ask questions. And so the tool that I'm working on for my research is a tool that does a specific type of this annotating terms to ontology, or annotating ontology terms to text. And what that looks like is we want to be able to take sentences representing phenotypes, like plants are shorter than normal, and translate them to a format that uses 2 different ontology terms. One term called the entity, which is shown in orange, which is describing the structure or function of the phenotype, and then a second term called the quality, which is shown in green, which is specifying how the entity is being modified. So we have whole plant, the entity, or E, being modified by decreased height, quality, the Q. And we consider this format of phenotypes easier to compute on because they have the properties of ontology annotations that we showed in the last slide, and they always have this E modified by Q structure. So one of the things that we want to use phenotypes that have been translated to this format for is to do candidate gene prediction. So I'll give an example on this slide of how that works. So what's being shown is 2 different mutant phenotypes. On the top, we have an increased branching phenotype in neurons in mice. And on the bottom, another mutant phenotype, which is showing increased branching of trichomes in Arabidopsis. And so whether you're thinking of these phenotypes as being described by the images or by the text that's describing them in the papers where they were reported, it's difficult to get a quantifiable measure of how similar they are. But when we do the translation to an EQ statement format, just like we showed on the first slide, we can look at the ontology subgraphs that are specified by those terms to get a measure of how similar they are. In this case, they both have the quality increased branching, so there's going to be a strong overlap. And we can say that these phenotypes across these 2 different species are equivalent in some way. Once we say that they're equivalent, say, for example, that we know gene A is the causal gene of this phenotype in mice, but we don't know the causal gene for the equivalent phenotype in Arabidopsis. What we can do is search the genome of Arabidopsis against gene A using sequence similarity or common pathways. And genes that are returned from that search, so in this example, gene B, are hypothesized to be involved in the phenotype in Arabidopsis. So a couple of different studies have demonstrated that this method using EQ statements to predict candidate genes works. But one of the issues is that because we're talking about thousands and thousands of phenotypes, each of those has to be, in the case of these studies, manually translated into an EQ statement format, which takes a lot of time and effort. And so in order to scale this method for finding candidate genes up to include all the unmined sources of phenotypes that occur in the biological literature, we need a method to automate this process of going from text to EQ statements, which is the function of the tool that we're working on. So this slide is going to be an overview of the methods that we're using in the pipeline to do this automation. So we start with the input. Going with the same example, it's just a sentence describing the phenotype. And the first question we ask is, which terms from some set of input ontologies simply match this text in some way? So what we're looking for is just an unstructured set of ontology terms that somehow map to this text. And we use an ensemble of different methods to accomplish this, which I'll go into more detail in a moment. Once we have this unstructured set of ontology terms, we apply a second step of the pipeline, which is to figure out which of those terms should be combined into an EQ statement, which makes the most sense given what the structure of the original text was. So we're looking to find an entity quality pair. And the way we do this is to use a natural language processing analysis of the original text and then test each EQ statement potential pair from our unstructured set against it to see which one fits the best. So it's a two-step pipeline, step 1 and 2, and I'll first go into more detail on the first step and then the second. So the first step, again, is to go from a sentence to an unstructured set of ontology terms, which is shown on the bottom. And the methods we use to accomplish this can roughly be grouped into two separate categories. On the left, we have some untrained algorithms. And by untrained, we just mean non-machine learning. And so these look for instances of ontology terms within text by accounting for things like variations in spelling, variations in synonyms that are being used to describe the same words, even misspellings, for example. And then on the right, we also employ some machine learning algorithms to do the same task. But these account for a wider variety of potential similarity between terms and the text that they might be being used to represent. And what we are left with after this step is just a set of terms that match the text. And they each have a score assigned to them based on how well they match the text, as well as which word within the text those terms match to. So we can evaluate the performance of this step simply by looking at how well the pipeline captured the terms that a human curator assigned to that same input text that was available. And that's what's being evaluated using a standard precision recall curve here. But what we're also interested in is when the pipeline predicts a term which is not exactly the same as the term that was used by the human curator, we want to know, was that term close to the term that the human picked, or was it very different in the context of the ontology? And so what the dashed line is representing is the average similarity between those false positives that the machine produced. And we can compare that to the average similarity that would be expected if the predictions were random. And we can see that it's higher. And then a similar type of analysis holds true doing this for entity terms, in addition to just qualities. So again, here's an overview of the pipeline, and now we'll look at that second step, which takes a set of unstructured ontology terms and tries to build a coherent EQ statement out of them. The way this works is we first use some natural language processing techniques to build what's called a dependency graph of the original sentence. And what that looks like is each node in the graph is a word, and then the edges that connect those nodes are grammatical or linguistic dependencies or relationships between them. And from the last step, each of the terms in our set is mapped in some way to one or more of those words. So what that means is that each term that we have to work with is mapped to a single node within the dependency graph. What that allows us to do is look at a possible EQ statement we could build and then evaluate it based on the relationship between the nodes that it maps to. So as an example, if we take short day photoperiodism as our entity and decreased height as our quality, those both map to the same node, which is shorter in the graph. And it turns out that a length of 0 between the nodes representing the entity and the quality occurs with a low probability. So we give that EQ statement a low score, which makes logical sense to us. If you swap out the entity for a whole plant instead, these map to two different nodes, plants and shorter. Those are connected by a length of 1, which occurs with a high probability. So we would give that EQ statement a higher score. So in general, we start with text statements. Those are the input to the pipeline. They're run through these 2 steps. And then the EQ statement that receives the highest score is the one that is outputted. We're calling that the predicted EQ. And we can compare those to curated EQs, which are the EQs that were generated by humans. And what this graph is showing, it's a histogram of the similarities between the predicted EQs from our method and those generated by humans. So instances to the left are where the prediction was very different than the human, and the ones further to the right are where they were more similar. And one beneficial property of the methods we've developed so far is that the ultimate score that we talked about in the last slide that these EQ statements receive, so this is the score that's output by the pipeline to say how confident the computational method is that the EQ statement is good. That score is roughly correlated with the similarity we find between the predicted EQ and the curated EQ. So if we put a threshold in place, like for example, if we want to get rid of all the predictions that scored less than 0.7, then we would remove all the predictions that are in these white boxes, which are poor predictions further to the left. So to summarize, we're working on tools to tap this wealth of phenotypic data that's available in databases and the scientific literature to translate them to the EQ statement format, which is a more computable representation of phenotypes that we can then apply towards generating candidate gene predictions. So I want to thank other members of my lab group and my PI, Carolyn Lawrence Still, the P3 grant for funding, and as well as the Plant Science Institute Scholars Program for funding. So thank you, and I can take any questions.
Awesome. Do we have any questions for Ian?
Where do you get the phrase data that you're using for training?
Currently, we're getting them from database entries that were involved in the study that we're using to compare the performance of our pipeline against. So it's a set of statements that have already been annotated by human curators or for a specific online resource called Plant PhenomeNet, but it's combined information from 6 different databases.
Okay, so are you training your system to extract these phrases?
That's something we're working on right now as a second portion that would go before the steps that I described. So it would look at journal articles and then pick out the phenotype descriptions from them as an additional step. Yeah.
Great, thank you.
Thanks.
What other questions do we have? Awesome. All right. Well, then with that, let's thank Ian for his talk and his